Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in genetics. Today in this session we learn about interaction of genes. In that we learn complementary gene interaction. Before moving on to the complementary gene interaction, first we should know what is interaction of genes. Mendel, by his experiment, concluded that each character is controlled by a gene or factor. But from recent investigation it is found that many genes affect the same character. Single character is controlled by more than one gene, and very often the phenotypic expression of an individual is the result of interaction between two or more genes. And Mendelian ratio of 3:1 and Mendelian ratio in dihybrid cross, that is 9:3:3:1, do not occur in all cases. The phenotypic ratio in F2 depends on phenotypic ratio in second filial generation, F2 generation, depends on the number of genes and type of interaction. The single gene, single character hypothesis is not always correct. This is known as interaction of factor or interaction of genes. The interaction leads to modification of Mendelian ratio, that is 3:1 or 9:3:3:1. Interaction of genes may be either qualitative or quantitative. The following are some examples of qualitative interaction between the genes or the factors, like complementary gene interaction, supplementary gene interaction, and epistatic gene interaction. In that we learn in this session about complementary gene interaction. The best example for complementary gene interaction is the flower color in sweet pea or Lathyrus sativus. In sweet pea plant we have two type of flower color. One is white flower and another one is purple flower. Here, when two white flower bearing plants were crossed, when two white or colorless flower bearing plants were crossed, so both the parents are the white or the colorless, the flowers bearing plants, and their genotype is small cc and capital pp. Similarly, the another flower, white flower bearing plant genotype is capital cc and small pp. Here, due to reductional division, there will be reduction in the chromosome number, and the parental gametes will be having 1 C out of 2 cc and 1 P for 2 P. So that means it is homozygous for PP and homozygous for small cc, or it is recessive. Here also it is capital C, single C out of 2 cc and 1 small p out of pp, homozygous recessive condition. When these F1 plants, parental plants were crossed, that is white flower bearing plants, in F1 generation all the plants were bearing flower color was purple. Here you should remember, when white flower bearing plants were crossed in F1 generation, all the plants were purple flowered. When F1 plants were selfed, when two purple flower bearing plants were selfed, So the genotype of the F1 plants were capital C, small c, capital P and small p. So here you can witness heterozygous condition for the chromogen and heterozygous condition for the product. So CC and capital P, small p. Here also it is condition because it is F1 generation. When F1 plants were selved, the F1 plants, they produces 4 type of gametes: capital C, capital P, capital C, small p, small c, capital P and small c, small p. Here also the another F1 plant will also produce same type of the gametes. The fusion of the gametes was represented on the Punnett square. So one plant was considered as male and its gamete was represented on the top, and another was represented as female, was represented here. When capital C, capital P carrying gamete fuses with the capital C, small p gamete of the male, it results in capital CC, capital PP, and the phenotype was purple. Similarly, when capital C, capital P fuses with capital C, small p, here also homozygous dominant for CC, heterozygous dominant for P. Here also the flower color was purple. Similarly, here capital C, small p when fuses with small c and capital P, here also we will get capital C, small c and homozygous dominant for P, hence it is purple. Here in this, when capital C, capital P fuses with capital C, small p, the heterozygous condition for C as well as heterozygous dominant for P. Then also the flower color was purple. When capital C, small p fuses with capital C, capital P, again the flower color was purple. Here capital C, small p, when it fuses with another capital C and small p, the flower was having homozygous dominant for the C and it is double recessive for P. And here the plant flower color was white or colorless. Capital C, small p gamete, when it fuses with small c and capital P, the flower color was purple because it is heterozygous dominant for C and P. When capital C, small p fuses with another capital C and small p, the flower color was white because it is heterozygous dominant for P, C and homozygous recessive for P. Similarly, when small c, capital P gamete fuses with capital C and capital P, flower color was purple because it was heterozygous condition for C and homozygous dominant condition for P. And when same gamete fuses with the capital C and small p, the flower color was purple because it is heterozygous dominant condition for C as well as P. Then when this gamete fuses with small c and capital P, it is homozygous recessive for C and heterozygous dominant for P. Similarly, when this gamete fuses with capital C and small p, Flower color again it was white. Small cc homozygous recessive for C, heterozygous dominant for P. When the small c small p both are in recessive condition, fuses with capital C and capital P, we will get capital C small c, capital P small p, heterozygous dominant for both the C and P, hence we get purple flowered plants. When this gamete fuses with capital C and small p, here it is heterozygous dominant for C and homozygous recessive for P, hence the plant flower color is white or colorless. Similarly, here also it is homozygous recessive for C, heterozygous dominant for P, again it is white flower. At last it is having heterozygous dominant for C, homozygous recessive for P, again it is white or colorless. When purple flowered plants were selved, F1 purple flowered plants were selved, F2 generation plants showed 9 purple colored flower and 7 white color flower bearing plants. Here character is produced by interaction of two or more dominant genes situated on separate chromosomes which complement to one another. Bateson and Punnett found that there were two different white flower varieties of sweet pea. A cross between them produced in F1 generation all purple flowered plants. When F1 plants were selved and they were self-pollinated, F2 generation consisted of purple flowered and white flower bearing plants with a ratio of 9:7. That means 9 are purple flowered and 7 are white flower. And this is a modification of normal dihybrid ratio of 9:3:3:1. You are getting 9, but instead of 3, instead of 3:3:1, here we are getting the 9 purple flower. Instead of getting 3:3:1, here you will see the 7 plants, they are white flower bearing plants. That's mainly because 9 plants are having the C, capital C and capital P either in homozygous dominant condition or in heterozygous condition. Both capital C and capital P are present in these plants, 9 plants. That's why purple flower. And among the 7, any one of the factor, that is either capital C is there in homozygous dominant or heterozygous condition, or P is there in homozygous dominant condition. Not both of them are there in 7 plants. That's why we are getting 7 white flower bearing plants. Here the purple flower, here the formation of the anthocyanin pigment depends on the purple flower, or the purple color is due to anthocyanin pigment. The formation of the anthocyanin pigment depends on 2 independent factors or the genes. Both of these should be present, both capital C and capital P. Both of these should be present to produce the anthocyanin pigment. Absence of any one throws the machinery of anthocyanin formation out of order. One of these factors, capital C, produces the colorless base, chromogen, while P produces an enzyme which activates the chromogen to produce the color. Here you have the C. The C produces chromogen. C produces chromogen, whereas the P produces the enzyme which acts on the chromogen and it produces the anthocyanin or the purple color in the flower. Anthocyanin will be produced. So this anthocyanin is due to the presence of both the chromogen and the enzyme. Absence of any one of the dominant factor throws the formation of the anthocyanin pigment. If there is no C, the chromogen is not there for the enzyme to act, and hence white flower. If there is no E, second step, that is the action of this enzyme on the chromogen, will be blocked. So C and E act as complementary to each other's function to produce color. They should be present in homozygous condition, capital CC, capital EE in dominant condition, or in heterozygous dominant condition, that is capital C small c, capital P small p. The two true breeding white races do not produce purple because each of them have either capital C or capital P and not both. If in F1 generation, capital C and capital P come together to produce the color. The white flower plants should contain the recessive alleles of other genes along with one dominant gene. One white flowered plant has the genotype of capital C and small pp. The another has small cc and capital pp. And hence in F1 generation, when they were crossed, they produces the purple flower with the genotype of capital C, small c, capital P, small p. Here chromogen is there, it will be activated by the enzyme to produce the product. When F1 plants were selfed, we get 9:7 ratio. 9 plants have either capital C and capital P in homozygous dominant or heterozygous dominant condition, and 7 plants have either they have the capital C or either they have the one capital P, not both. That's why we get 9:7 ratio. The genes capital C and capital P are complementary genes. They can be defined as two or more non-allelic separate dominant genes present on the different loci interacting to produce a phenotype, a novel phenotype, neither of which can produce that trait alone. They cannot produce the novel purple flower alone. The combination of complementary genes explain the phenomena of throwback or reversion to an ancestral type. The similar inheritance occur in animals and also in man. Some traits like exceptional musical talent, high intelligence may result from the interaction of complementary genes, each coming from different parents. This may explain the fact that sometimes extraordinary ability appear in one member of the family while other children and parents are not outstanding. That is called reversion or throwback. I hope you understood this complementary gene interaction To express the novel character, both the dominant genes should be present either in homozygous dominant condition or in heterozygous dominant condition. It's in a complementary, they both the dominant gene, they complement to produce the novel character. I hope you understood this complementary gene interaction. It's a type of interaction of gene. Thank you.